Welcome. In this lecture today, we will learn about antigen recognition by the immune system. Our immune system recognizes antigens by the B cells and the T cells. B cells recognize antigens by the receptors present on their surface, also called the immunoglobulins or the B cell receptors. So today we will learn about these B cell receptors or the antibodies with special reference to the monoclonal antibodies, how they are produced and what are their functions. Immune system also recognizes invading antigens by the T lymphocytes. Today we will also learn recognition of antigens by T cells with special reference to the MHC molecules or the major histocompatibility complex molecules. What is an antibody? An antibody or the immunoglobulins are the proteins produced by our immune system upon invasion by antigens. An antigen is a foreign protein which is not supposed to be normally present within our body. And antibodies which are produced upon invasion by an antigen are glycoproteins in nature, and they belong to a big class of protein groups called the immunoglobulin superfamily proteins. Antibodies can occur in two physical forms. Either they are present as secreted forms in the blood plasma, and they are secreted by a very specialized cells called the plasma cells, or they occur as surface proteins on the B lymphocytes, and in this form they are called the B cell receptors. This slide shows the basic structure of the antibody molecules. Antibody molecules are made up of 4 chains, 2 heavy chains and 2 light chains. These heavy and light chains are joined together by disulfide linkages. Globally, the structure of the antibody molecules are more or less same, but they have minute difference in their variable region. And the region that is overall same with many different antibodies are called the constant regions. If we go into the detailed structures of the antibody, we see that the heavy chain can be further divided into 4 distinct domains called the variable domain and the 3 constant domains named as CH1, CH2 and CH3 as shown in blue in this figure. CH stands for constant region of the heavy chain, and antigen binding by this antibody takes place at the tip of the Y-shaped molecules, where the variable regions of the heavy chain and the variable region of the light chains are present. In this diagram, you can see there is a hinge region shown in green. The hinge region allows the prongs of the antibody molecule to stretchen or shorten so that it can accommodate nicely and finely the invading antigen molecules. The CH2 and CH3 domain comprises the FC region of the antibody, which means it's called fraction crystallizable because this part of the antibody molecule could be crystallized. And the VH domain, CH1 domain and the light chain comprises the FAB domain of the antibody. FAB stands for fraction antigen binding because this is the region of the antibody where the antigen binds. Normally, one antibody molecule can bind to 2 antigen molecules at its 2 prongs. That's why antibody molecules are usually called to be bivalent for antigens. Antibody molecules are classified depending on the type of heavy chain they possess. And there are 5 different types of heavy chains possible. They are termed as alpha type, gamma type, delta type, epsilon type or the mu type. And there are 2 types of light chain possibles, either kappa type or lambda type. And depending on that, immunoglobulins or antibodies are classified as IgA when the heavy chain is alpha type, IgD when the heavy chain is delta type, IgM when the heavy chain is mu type, and so on. And in all the 5 cases, the light chain can be either kappa type or lambda type. It's also worth mentioning that IgA can exist in dimeric form and IgM in pentameric form. Now, when an antigen invades, at first it binds to the antibody molecules that are present and projected on the surface of the B cells. Binding of antigen to these surface bound B cells per se have no direct immunological consequences, except this triggers development and proliferation of the B cells. And these B cells then differentiate, sometimes getting signals from the helper T cells to become plasma cells, and these plasma cells secrete copious amount of immunoglobulin molecules into the blood, and these secreted antibodies then fight against the invading antigen. It can neutralize the antigen, it can precipitate the antigen. Now, whenever a foreign antigen invades, each antigen can have several epitopes onto which different antibody molecules can bind. This leads to what is called a polyclonal response. That means even if there is only one antigen which can contain many epitopes, at its different epitopes, different B cells can bind.
Sir, I have a question. What is the fundamental difference between a polyclonal and monoclonal response?
Whenever an antigen invades in a whole organism, and as I told you that an antigen, suppose it's a whole bacteria, it can have several epitopes. So to each of these epitopes, different antibody containing B cells comes and binds. And then all these B cells that got a chance to bind to the different epitopes of this single antigen will proliferate to produce plasma cells. So after a couple of days, the plasma of that particular whole animal will be rich in antibody molecules produced by different B cells. This is called a polyclonal response, which is actually beneficial for the host. But the problem is there is no homogeneity in the antibody population. In contrast, monoclonal antibodies are monospecific antibodies. They are produced from a single type of B cells and they recognize a single epitope. That's why in a monoclonal antibody population, all the antibody molecules are identical. That's why the cross reaction is less. Normally, monoclonal antibodies are not produced in a whole animal. Monoclonal antibodies has to be produced with very sophisticated, precise techniques which will be discussed today. Okay? Normally, in a mammalian cell, DNA is produced by 2 pathways. Either they are produced by de novo pathway, where phosphoribosyl phosphate and uridylate are required, and they ultimately produce IMP, from which DATP and DGTP are produced, which are used as the DNA bases. There is a second pathway called the salvage pathway, where hypoxanthine converted to IMP and thymidine is converted to DTMP, from which the nucleotides are produced. 2 very important enzymes are required for salvage pathway. To convert thymidine to DTMP, thymidine kinase is very much crucial an enzyme, and HGPRT or hypoxanthine guanine phosphoribosyl transferase is a very key enzyme for conversion of hypoxanthine to IMP in the purine salvage pathway. The technique with which monoclonal antibodies are produced in laboratory conditions is called the hybridoma technology, where at first a mice is inoculated with the test antigen, meaning the antigen against which we want to develop the monoclonal antibody. There is a definite elaborate process. The mice is first injected with the antigen, then after 14 days and 28 days it receives booster doses, and then the mice is sacrificed and the spleen cells are harvested. Then the spleen cells are fused with myeloma cells. What are myeloma cells? Myeloma cells are B cell tumors, and these cells are immortalized, they can grow indefinitely in tissue culture setup. For hybridoma experiments, myeloma cells with specific genetic background are taken. As you can see in this slide, they are HGPRT minus, meaning they are deficient in hypoxanthine guanine phosphoribosyl transferase, which I showed is a key enzyme for the purine salvage pathway. Meaning these myeloma cells cannot produce DNA bases through salvage pathway. They have to rely on the de novo pathway. When these two cells are fused together either with PEG or any other fusing agents, heterokaryons are formed, and when they replicate, they gradually lose chromosomes. And then we have to test for the positive clones. Now there is a question. The frequency of fusion forming hybridoma is very less, like 1 in 10 to the power 5 or even lower. So whenever you try to do hybridoma, there are always unfused parental cells. That means the spleen cells of the mouse and the myeloma cells that are left. Those are not your desired cells. You have to get rid of them. And for that, there is a selection. It's called the hat selection. How the hat selection works? In the growth medium, after the hybridoma is made, they are allowed to grow in a growth medium which is called the hat medium, where hat stands for hypoxanthine, aminopterin and thymidine. As you have seen in my previous slide, aminopterin is a blocker of the de novo pathway of nucleotide biosynthesis. So in the hat medium, the unfused B cells normally die because they have a limited lifespan. They are differentiated cells. And the myeloma cells also die because they are deficient in HGPRT, so they cannot use the purine salvage pathway. And in the medium there is aminopterin, so the de novo pathway is also blocked for them. So both the pathways of nucleotide synthesis is blocked for the myeloma cells. That's why they die. But the hybridoma cells survive because they get the immortalized property from the myeloma cells, and they get the nucleotide biosynthesis option through the normal B cells, where the salvage pathway is still functional. And in the medium, thymidine and hypoxanthine is present. Now at this point, the surviving hybridoma cells has to be tested for the antibodies against the antigen with which the mouse was inoculated. The positive clone has to be chosen, and now this positive clone can be expanded or maintained in tissue culture setup for production of copious amount of monoclonal antibodies, or with these positive clones, tumors could be induced in mice, and then this tumor can once again be the source of monoclonal antibodies. In the year 1984, Georges Koholar, Cesar Milstein and Niels Katjärni received Nobel Prize for mastering the technique of making hybridomas and production of monoclonal antibodies. Uses of monoclonal antibodies: diagnostics and ELISA test kits for AIDS and many other diseases, and the simplest of the pregnancy test kits which use monoclonal antibody against human chorionic gonadotrophin. Monoclonal antibodies can also be used for treating viral diseases which are otherwise very difficult to treat. Pathogenic bacteria can be serotyped and classified using monoclonal antibodies. As you know that pathogenic bacteria contains very precise antigenic determinants on their surface. Since monoclonal antibodies don't have any cross-reactivity, they can be used for detection of specific serotype of a particular bacteria. Monoclonal antibodies have unending uses in biomedical research. Monoclonal antibodies can be used for detection of minute amount of any proteins in biological setup for ELISA or radioimmunoassay type of experiments, in organ transplantation also, and once again I tell you, there can be many more roles and uses of monoclonal antibodies. MHC molecules were first identified as being responsible for graft rejection in mice experiments. Later on, it was seen that MHC molecules have even more important roles to play in immunology, because the fundamental difference between recognition of antigen by B cells and T cells is the fact that B cells can recognize soluble antigens, but T cells recognize antigen only when it is properly presented through these MHC molecules. MHC molecules, or the major histocompatibility molecules in humans, are called human leukocyte antigens because they were first identified on leukocytes or the white blood cells. And in mice, these molecules are called the H2 antigens. If we go to the structure of the MHC genes, we see that the genes of MHC are located on the short arm of human chromosome number 6, spanning a long range of 3.5 million base pairs. The structure of both MHC class 1 and class 2, we will see that there are 3 classes of MHC molecules, class 1, class 2 and class 3, of which class 3 are of little importance. We will mainly focus on MHC class 1 and class 2, and the crystallographic structure of MHC class 1 and class 2 are solved. MHC class 1 molecule is expressed on all nucleated cells except some nerve cells and platelets, and MHC class 2 molecules are expressed on antigen presenting cells or the AP cells like the lymphocytes, the dendritic cells, the macrophages, the monocytes, Langerhans cells and so on. And cytokines, especially the interferon gamma, can increase the level of expression of MHC1 and MHC2. Now HLA typing is very important. As you have seen that there can be HLA A, B and C, and also for class 2 MHC there can be DP, DQ and DR. HLA typing of an individual is actually the signature of its own genetic makeup. HLA typing is also very important during the organ transplantation between allogenic siblings. Class 1 HLA typing is mainly done by microtoxicity assay, and class 2 MHC typing is mainly done by PCR and sequencing methods. There are several diseases that are related to MHC molecules, like the diseases associated with class 1 MHC molecules are ankylosing spondylosis, psoriasis vulgaris or Ritter's disease. These are mostly of autoimmune origin. For example, in ankylosing spondylosis, the marker B27 is found to be highly expressed. Similarly, for type 2 MHC diseases, there is Hashimoto's disease, which is the autoimmune thyroiditis. There is multiple sclerosis, rheumatoid arthritis, insulin-dependent diabetes, and so on. So what is the structure of the class 2 MHC molecule? It contains 2 non-identical chains called the alpha chain and beta chain. Each of them have 2 domains, namely the alpha 1, alpha 2 and beta 1 and beta 2. Except for alpha 1 domain, all the other 3 domains, that is beta 1, beta 2 and alpha 2 domain, they are stabilized by disulfide interactions. Alpha chain is 34 kD and beta chain is slightly smaller, 28 kD. The peptides that are facing the alpha 1 and beta 1 domain are highly polymorphic. The other part of the molecule is more or less constant, and in this polymorphic region the antigen is presented. Alpha 2 and beta 2 is more or less constant and non-polymorphic. The peptides that these class 2 MHC molecule presents are 13 to 25 amino acid residue long. It's important to note that the source of class 2 MHC molecules are exogenous antigens. When the exogenous antigen is endocytosed, then they are fused with lysosomes where the acid hydrolases, they cleave and process these antigens. Then they get folded along with the alpha and beta chains of the class 2 MHC molecules, and then things get properly folded, and then they are expressed and presented on the surface of the APC cells where CD4 positive T helper cells interact. The structure of the class 1 MHC molecules. Class 1 MHC molecules also contain 2 chains. One is heavier, one is the lighter. The heavier chain is 44 to 47 kD in size, whereas the smaller chain, which is called the beta 2 microglobulin, is only 12 kD in size. The heavy alpha chain contains 3 domains called alpha 1, alpha 2 and alpha 3. Alpha 2 and alpha 3 is stabilized by disulfide bonds, but there is no such disulfide linkage in alpha 1 domain of the heavy chain. Both alpha and beta 2, these molecules also belong to the global immunoglobulin gene superfamily molecules. They have structural resemblance to the immunoglobulin superfamily molecules, and the peptide they present is 8 to 10 amino acid long. And similarly, like the MHC class 2 molecules, here also the antigen is presented through the alpha 1 and alpha 2 subunit of the heavy chain, and the regions that face the antigenic site of the alpha 1 and alpha 2, you see polymorphism, which makes every MHC class 2 molecule distinct from the other. And these MHC class 1 molecules, they present antigen to the CD8 positive cytotoxic T lymphocytes. Source of antigen for class 1 MHC molecules are endogenous antigens. It can be a viral antigen. These antigens are sensed as foreign by the cellular system, and then they are degraded and processed by the proteasome the way other mutant cellular proteins are handled. Then once again, they are co-folded with the alpha and beta of class 1 MHC molecules, and then they are projected onto the surface, which is further then recognized by CD8 positive CTL. In 1980, John Dosset, George Snell and Boruj Benacerraf, they received Nobel Prize for their work on antigen processing and presentation by MHC molecules. I conclude by showing this slide that there is a fundamental difference between antigen processing and presentation and in terms of source of antigen by type 1 and type 2 molecules. Type 1 MHC molecules accept endogenous antigens, be it a viral protein, be it a defective cellular protein, then it goes through the proteasome degradation pathway and ultimately co-folded with the alpha beta part of the MHC1 molecule, and then they are projected on the surface of the T lymphocytes. In contrast to type 2 MHC molecules who accept only endocytosed exogenous antigens, which is processed and then produced and presented with the type 2 MHC molecules for the CD4 positive helper T cells.